Good morning, students. Today is the third day of chapter Human Reproduction, plus 2 class Biology. In the previous day 1 and day 2 lecture, we have already covered about the events of reproduction, about the male reproductive system, and then we started the female reproductive system in the previous day 2 topic. So, start our first topic. This is the you can say continue part of day 2 lecture. This is a sectional view of female reproductive system, which is already covered. And the female reproductive system consists of fallopian tube and a pair of ovary and the layer of uterus, endometrium, myometrium, perimetrium, cervix, cervical canal, and vagina. Already covered in the previous day 2 lecture. So, and the first topic of today's lecture is accessory ducts. Accessory ducts. What are the accessory ducts? Accessory ducts are, you can say, which help in the reproduction. Accessory means there are two type of functions in our male or female reproductive system. First function is to produce gamete, means to produce the male gametes and female gamete. Male gamete is produced by, yes, testes, and the female gamete is produced by ovaries. And the second function of reproductive system is to transfer or to transport the gamete from the male or female reproductive you can say organ, that is testes and ovaries, to the site where fertilization take place. Means accessory ducts are the ducts that provide the passage for the transfer of gametes. So in female accessory ducts, oviduct, uterus and vagina is there. Oviduct is the fallopian tube. Fallopian tube is near about 10 to 12 centimeter in length, and it will start from the ovary and it will join with the uterus. The fallopian tube contain a finger-like projection known as fimbriae. I am mentioning with the arrow. This is the finger-like projection here, fimbriae. Fimbriae is to collect the egg that is released through the ovary. Then infundibulum area is there. Then ampulla area is there. Then isthmus is there. So these are the different you can say parts of a complete fallopian tube or a oviduct. And the fertilization or the site of fertilization is ampulla isthmic area. Ampulla isthmic area is the site of fertilization. Then uterus. Single uterus is present in you can say female body, and the lower area, or you can say the complete uterus, is known as womb, and it is a hollow inverted pear shape area. Hollow inverted pear shaped structure means uterus is a inverted pear like structure, and it is attached to pelvic wall by the ligaments are present which connect the, or which attach the uterus. Then inside the uterus, fertilized ovum means when the ovum, when the egg gets the female gamete. So inside the uterus, fertilized ovum grow and develop into a embryo, and this will open into a vagina through a narrow canal known as cervical canal. Cervical canal is the narrow area, and cervical canal along with the vagina, then it will termed as birth canal. And the wall of the uterus contain three layers already discussed: perimetrium, myometrium and endometrium. Perimetrium is the outermost layer of uterus, then middle layer is myometrium, and it will contain the smooth muscles. And during the delivery, during the childbirth, the myometrium muscles start contraction, and endometrium is the innermost layer of the uterus, and endometrium contain the glandular layer. Granular layer means endometrium contains some glands, and these glands release some type of secretion, you can say. And endometrium is the layer which undergo the cyclic change, during menstrual cycle. During menstruation cycle, this endometrium layer get break down, and it will lead to release of some type of waste or blood from the female body to the outside. So next, now external genitalia. External genitalia contain the mons pubis, labia majora, labia minora, and hymen and clitoris. This is covered in the, you can say, pubis, the first slide of part 2. What is mole pubis? Mole pubis is the fatty tissue covered with the hair. Means female external genitalia contain a fatty tissue covered with the hair, name known as mons pubis. Then labia majora. Labia majora is the fleshy fold of tissue surrounding the vaginal opening. Means vaginal opening contain surrounded by a fold of tissue known as labia majora, and inside the labia majora there are also a paired fold of tissue known as labia minora. And then clitoris. Clitoris is the finger-like structure. Clitoris is the finger-like structure. It is present in the upper area, upper junction of two labia majora. Then hymen. Hymen is present just inside the opening of vagina. Hymen is the area, or hymen is the structure or the layer which is present just inside the opening of a vagina. Hymen is a thin, you can say, covering, and it is oftenly torn during the first mating. When first mating take place, hymen gets ruptured. Next, the presence or absence of hymen is not the reliable indicator of virginity. Sometime hymen get accidentally tear or torn up during some physical exercise, during cycling, or during swimming. So hymen is not the confirmatory, you can say, indicator for the virginity of any female. Next is mammary gland. Mammary gland is the accessory gland that is present in female reproductive system. Mammary gland is paired in structure, and mammary gland contain glandular tissue, and it contain the fat tissue. Glandular tissue divided into 15 to 20 mammary lobes, and mammary lobes contain a cluster of cells known as alveoli. Alveoli is secrete the milk. Alveoli secrete the milk, and this milk is stored in the lumen. Lumen means space. Lumen means space inside the alveoli. Alveoli open into mammary tubules, and mammary tubules combine to form mammary duct, and these duct combine to connected with the lactiferous duct through which milk is sucked out. You can see these are some fat storing tissue. Then mammary lobe is there, mammary alveolus, mammary duct, ampulla, this one, lactiferous duct, and through lactiferous duct milk is sucked out. This is the structure of mammary gland. And here only one point is important. Milk production take place in alveoli present in mammary gland. Now next, gametogenesis. What is gametogenesis? Gametogenesis is the process of formation of gamete, or you can say the process of formation of haploid gamete from diploid germ cell is known as gametogenesis. There are primary sex organ are present in male and female both. The primary sex organ in male is testis and the female is ovary, and the function of testis is to produce sperm, and the function of ovary is to produce ovum. The process of formation of sperm is known as spermatogenesis, and the process of formation of you can say egg is known as oogenesis. And there is an interesting difference between male and female reproductive system. The sperm formation take place when the male get mature. Means when the male is mature, and or when the male get attain the puberty age, then the sperm formation will take place. But in the female, the formation of egg starts at the embryonic stage. But in the female, the formation of egg will take place at the embryonic. When the female is in fetal stage, in the fetus area, embryonic period. So during embryonic stage, the formation of female gametes start. But in the case of male, the sperm formation will take place when the male achieve the puberty age. Now, first is spermatogenesis. What is spermatogenesis? Spermatogenesis is the process of formation of sperm or haploid male gamete, and the site of sperm formation is seminiferous tubule, already covered in the male reproductive system. Seminiferous tubule is present inside the testes. So, seminiferous tubule is the area where spermatogenesis take place, and the inner wall of seminiferous tubule contain two type of cell already covered: the germ cell or spermatogonia cell and the Sertoli cell. And the germ cell divide and develop into the sperm, and Sertoli cell provide the nourishment to developing sperm. So these are the some points regarding spermatogenesis. Next, this is the complete process of spermatogenesis. Spermatogenesis, you can say, divided into three main substages. First is multiplication and second is growth phase. You can see here spermatogenesis is explained in the flowchart form also. This is the testis, and through testis spermatogonium is present. Spermatogonium is the cell that will lead to formation of sperm, and spermatogonium will change into the primary spermatocyte. Primary spermatocyte contain diploid chromosome. Here 46 chromosome is there, and in primary spermatocyte first meiotic division take place. First meiotic division take place means meiosis will take place, and it will lead to formation of secondary spermatocyte, and it will lead to formation of secondary spermatocyte. And secondary spermatocyte is haploid in state. Haploid means single set of chromosome. Primary spermatocyte is diploid in set. Diploid means 46 chromosome is there, but in secondary spermatocyte haploid set is there, 23 chromosomes are there. So then the secondary spermatocyte will change into spermatids, and by further structural changes sperm gets formed. So let's start with the multiplication stage. In multiplication phase, the spermatogonia undergo the mitotic division. It is an important point. You must note that in multiplication phase mitotic division take place. Mitosis means equational division will take place, and it will lead to formation of spermatogonia cell which contain the same number of chromosome, that is 46 chromosome is present. In growth phase, the spermatogonium cell increase in size, and it will form the primary spermatocytes. Primary spermatocytes also contain the same set of chromosome, that is 46 chromosome is there. Next phase is maturation phase. In maturation phase, the diploid primary spermatocyte undergo first meiosis. You should note this word, that that first meiosis take place in maturation phase. And in maturation phase, the diploid primary spermatocyte will change into haploid cells known as secondary spermatocyte. Secondary spermatocyte is haploid in number. Secondary spermatocyte is haploid in number. Then second meiotic division take place, and then second meiotic division will lead to formation of four haploid spermatids. Four sperms get formed. Next is spermiogenesis. Spermiogenesis is the you can say formation of spermatozoa or formation of sperm from non-motile structure. Means when spermatids are transferred into the motile sperm. Motile means movement. Motile sperm is known as spermiogenesis. And after the spermiogenesis, means after the sperm gets formed, the sperm will release. The sperm will move out from the seminiferous tubule. All the events that are described earlier will be in you can say seminiferous tubule area, seminiferous tubule structure. And when the sperm get fully matured, then the sperm will move out through seminiferous tubule. This is known as spermiation. So this is all about in today lecture of human reproductive system. Thank you.